We'll start just by talking a little bit about the cell membrane, its structure, and what it functions as. So in eukaryotes, the cell membrane, also known as the plasma membrane, is comprised of a phospholipid bilayer with other lipids and proteins situated around and integrated into the bilayer. So bilayer, obviously, we have two layers that are on top of each other, and it's created of phospholipids, which are a little different than the triacylglycerols or triglycerides that we've been learning about, which we will talk about as well. So the membrane's primary function is to regulate the entry of materials into the cell. The eukaryotic cell membrane is considered semi-permeable because it is selective as to what may enter. So generally, only small nonpolar molecules can diffuse through the membrane without the help of a membrane protein. So any larger molecule or any polar molecule can't really get through that membrane too well, just because of that hydrophobic domain that we have in between the lipid bilayer. There are, however, ways that we can get these large protein molecules through that lipid bilayer, and we use a number of different proteins, transporters, channels that are in the membrane, integrated into the membrane that help. So that all of that is what combines and gives the cell membrane its sort of semi-permeable characteristic. Okay, so a closer look at the phospholipid bilayer. You could look at the right. Right here, the extracellular and intracellular. These would just be the environments regarding the cell on either side of the cell membrane, and you could see how they're situated with those hydrophobic tails inside and those heads outside. So a phospholipid is a lipid that is comprised of two fatty acids, chain tails, and a polar phosphate head group attached to a glycerol molecule. And think about triacylglycerols. So when we were learning about that, triglycerides, we have that choline group, that head group, and then there are three fatty acids that are attached to that choline group. So we still have those three bonding domains, but in the phospholipid bilayer, only two of them are taken up by fatty acids, and that third one is actually attached to a phosphate group. And yeah, so that's how we get these phospholipids that make up the cell membrane. So the cell membrane consists of two back-to-back phospholipid bilayers arranged with their hydrophobic tails pointing inward and their hydrophilic heads pointing outward. Why is this? If you're not sure, it's really just, it has to do with energetics. Shielding, energetics. The hydrophobic groups are obviously going to congeal and they're going to congregate together. And most of the extracellular environment and the intracellular environment, it's aqueous and it's charged. So these phosphate head groups are going to be pointing outward. Sort of like when we were learning about hydration shells and how hydrophobic materials will act in an aqueous environment or in a hydrophilic environment. So we're basically minimizing the surface area of these two surfaces touching each other, these hydrophobic and hydrophilic surfaces. And in order to do that, this is the most energetically efficient form that takes place. The large hydrophobic interior created by the phospholipid tails prevents polar molecules from entering the cell, like we said before. Those large molecules and those polar molecules are not going to be able to pass through that hydrophobic tail, those two layers of hydrophobic tails, so they have a more difficult time getting into the cell. So the fluid mosaic model describes the membrane as a fluid mixture of phospholipids, cholesterol, and proteins. So if you know what a mosaic is, a fluid mosaic, you can just think about all these different components sort of just bobbing around in sort of an ocean of phospholipids and cholesterols, and they're just sort of bopping around, bumping into each other. And obviously that fluidity changes with temperature, for example. Colder temperatures, the fluidity of the cell membrane might go down, and in higher temperatures, the fluidity is going to go up, and that can affect a lot of the regular functioning of the cell. So cholesterol helps maintain the structure in extreme temperatures. So in really cold temperatures, cholesterol actually manages to keep the cell membrane more fluid, and in higher temperatures, it actually manages to keep the cell membrane a little bit more rigid. So we'll take a quick look at some of the membrane proteins that you find inside the membrane. Proteins found in the plasma membrane can be either integral or peripheral. So integral proteins are integrated through the entire membrane and reach both sides. They can act as receptors, transporters, channels, enzymes. Peripheral proteins are only exposed on one side of the membrane, and they may act as enzymes, carriers, or anchoring proteins. So remember, peripheral proteins can't really be transporters or channels because they're only touching one side of the membrane. The whole point of a transporter usually would be to use ATP or some sort of energy to move some sort of substance, usually against its concentration gradient. And then channels are just an open channel where a substance can flow down its concentration gradient. And we can't do that with peripheral proteins because there's no connection between the internal and external environment. So keep in mind that while many surface receptors are used to pick up hormones from the bloodstream, cells may also use these proteins to communicate with surrounding cells. So I just added that just to give you an idea of the diversity of proteins. They're not just all transporters or channels. Some proteins do have enzymatic activities on the front. Some are used as receptors, and some even have proteins that are sticking out that can be used as ligands for the receptors of other cells. That's one of the ways that cells interact with each other. They may also have receptors for paracrine signaling from surrounding cells. They may release some sort of ligand that the cell can use to start some sort of signal cascade from a close cell. That's what we call paracrine signaling. Okay, this takes us to the first review question. So which of the following substances would most likely be able to diffuse through the plasma membrane without the help of a transporter protein? So think about everything we were just talking about, your knowledge of everything below. Take about a minute. I would pause the video because I'm going to go over the answer in about 5 seconds. So the correct answer would be D, carbon dioxide. And let's take a look at it. So A, inorganic phosphate. If you remember inorganic phosphate from biochemistry or orgo or any biology at this point, it's a pretty big polar, it's not huge, but it's a polar molecule, and that's the most important part. It's not going to be able to get through that hydrophobic tail region of the lipid bilayer. So inorganic phosphate most likely would need some sort of protein to get through. Serine, the same thing. Serine is an amino acid, and it has a hydroxyl group on its R group. So it is pretty polar, and again, it would have trouble getting through that hydrophobic domain. Palmitic acid, same thing. It's an acid. It's polar. It's pretty big. 16 carbon chain, if you remember. It's difficult to get through the plasma membrane because it's big. But D, carbon dioxide, is the best answer because it is the smallest non-polar molecule, and often carbon dioxide does diffuse simply in and out of the plasma membrane. So D would be the correct answer.